Welcome everyone. Welcome to Unboxing Biology with Smriti. In the previous video, we have discussed about the 7 contrasting traits which were taken by Mendel for his experimental work. Now, we will be discussing here in this video about the Mendel's laws. So, Mendel did experiment on the pea plant and gave 3 laws. The laws were known as law of inheritance. Okay. Now, the 3 laws name were, first was law of dominance. Second one is law of segregation. Third one is law of independent assortment. Okay. Now we will discuss here all the 3 laws. Fine. The first law is the law of dominance. So, Mendel stated that if we start with the pure lines means only the homozygous parents. Okay, I already told you the homozygous meaning is that when the alleles of a gene are both either dominant or the recessive. Okay, so we are starting here with the pure lines. Now, then Mendel actually stated that whenever we start with the pure lines, the only character or trait which is going to express itself is the dominant one. Okay, so for understanding this, we are taking one example. Let us take, we're taking the stem length character. This is a parent. You can say as a man or a male is crossed with a female which is homozygous or recessive. Now we will take out the gametes. Okay, so this is tall and this is the dwarf stem length. If you remember this, the dominant one is the tall and the recessive one is the dwarf. Now we are taking here the gametes. Okay, gametes, and we will now make the punnett square. Punnett square is defined as the graphical representation of all the genotypes possible of an offspring in a particular breed or a particular cross. Fine. So we will make this in the next slide. Let us make this. You just have to make a table like this. Okay. Now, let us take, it is the male here. This will be the female, and just make a divider. Now, write the gametes here. Like here it is capital T, small t are the gametes. So you have to write just like capital T, here the small t and the small t. I just forgot here to make one line. Let me make it. Okay. So now let us cross. Capital T with the small t will give capital T small t. Again the capital T with the small t will give capital T small t. Here also it will be the same. Here it will also be the same only. Okay. Now after making this punnett square, this is the F1 generation. How F1 generation is actually the breeding or the crossing between the parents. Fine, this is the F1. Whereas F2 is interbreeding between the offspring. Interbreeding between offspring. Okay. Now, here this table is the F1 table. Okay. Now you have to write about, I already told you, the phenotypic ratio. Phenotypic ratio means the phenotype means the physical appearance of an organism. So here the ratio will be, this is also the tall. In the phenotype we see the physical appearance of that organism. So here it is tall, 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 and fourth one is also the tall. Fine. So 4 tall and with the 0 dwarf. So the ratio will be 4 is to 0, where the 4 is tall, whereas 0 is a dwarf. Okay. Now the genotypic ratio, okay, the genotypic ratio obtained, this is the ratio, okay. So the genotype, I already told you meaning of genotype. It is the genetic makeup of an organism. So this is also heterozygous, this is also heterozygous, this one and this one are the heterozygous. So there are 4 heterozygous with no homozygous. That's why the ratio is 4 is to 0, where the 4 is heterozygous, okay, and none of them are homozygous. So, according to Mendel, it was stated that when we start with the pure lines, it is the dominant that expresses itself. Therefore, we can see here that the dominant is getting expressed. So, you can say that it is proven, hence, the character, or trait which expresses itself is the dominant one. Dominant one. Okay. Now we will study the second law. Second law is law of segregation. Okay, this is our segregation. Fine. Now, according to Mendel, first, this law is also known as law of separation, because during gamete formation, so during gamete formation, the alleles of a gene gets separated. Fine. So let us take example. So if we take capital T, let us take this example. So here, this is the homozygous example. You can say this is the homozygous one. So you can say here that the gametes here produced capital T and this capital T. Fine. Both are capital. Now, this is also, you can say here, or let us take something different. Okay. So we are taking here the capital T, small t, that is again the heterozygous. Fine. Heterozygous. Oh, I'm sorry. Heterozygous. Fine. Now, again the gametes which are produced here is the capital T and small t. Here, this gamete is pure for the particular allele. Fine. Now you can say all here, all the gametes which are produced are the pure for the particular allele. Fine. That's why, that's what this law wants to say. Therefore, this law is also known as law of purity of gametes. Fine. Now we will study the third law by Mendel, that is the law of independent independent assortment. Okay. Now, this can only be explained in the dihybrid cross. I'll tell you the monohybrid cross, dihybrid, and the trihybrid. Mono means single, di means two, and tri means three. So, when we do hybridization between the single character, it is the monohybrid. When we do with the two characters, taking the two characters, then it is dihybrid. When we take three characters, then it is the trihybrid cross. Okay, so here it can be explained only in a dihybrid cross, best explained. Okay, now we will just take an example first to know. Okay, wait, first you have to take a cross, like we are taking a capital T, that is the stem length, dominant one, and the color of the flower, that is the violet one. Fine. We are crossing it with the small t, that is the dwarf, and the small p, that is the white color flower. So we are taking two characters, tall and violet. Here it will be the dwarf and white. Okay, so these are the parents parents. Okay, now we will make gamete. So how you make gametes? This capital T is crossed with this capital P, then this capital T is crossed with this capital P. Now again this capital T with this P, and this capital T with this P. Okay, so we will obtain four gametes. TP, again T with this P, TP, this capital T with this capital P, capital TP, again the same. For this one, again we will get four gametes. You just have to make, or you just have to write, first explain it how I am writing this. We are, let me take another color to best explain. This small t is crossed with this small p, then this small t is crossed with this small p, then this small t with this p, and this small t with this p. Okay, so here we are getting, okay, so TP, if you cross it you will get these four gametes. Fine. So let me write here gametes. Now after making gametes, we usually make punnett square. Okay, I already told you what is punnett square. The F1 generation we are going to make it and make for this now. So okay, so I will make a table. This is okay. You just have to make a table like this. Okay, and because we have obtained the four gametes, that's why we will make the four rows and columns. One, two, three, and four. Okay, So now we will write on this. Let us take it as a male, and here it will be the female one. What were the gametes we obtained? So it was capital TP, the four here gametes, and the here are the four gametes. Okay, so we will write here also capital TP, capital TP, the same. I'm sorry, small p. Okay, now we will do a cross and make this Punnett square. Capital T, small t, capital P, small p. So we will write this like this. Okay, then this with this, capital T, small t, capital P, small p. Then this with this, capital T, small t, capital P, small p. Then again this with this, capital T, small t, capital P, small p. Okay, so here all the columns will be the same. Okay. Okay, now just fill this column as capital T, small t, capital P, and small p. You can just check anywhere, it will be the same. Capital T with small t, capital P with small p. Okay. Okay. Now we got these much offsprings, possible genotypes we got. Okay. Now we will just do selfing of the offspring. Selfing means we are doing the self fertilization here. Okay, so the possible genotype we got was capital T, small t, capital P, small p, and it will be crossed with the capital T, small t, capital P, small p. So first this capital T with the small p to just we want to get the gametes. So this is the parent. Now this offspring has become the parents because we are doing here selfing, that is the self fertilization. So now this is the parent, capital T. Okay, so this capital T with this capital P, then this capital T with the small p. Now let me change the color. Small t with the capital P, and again small t with the small p. So we will get again four gametes, that is capital T with capital P, that is okay. Now this is the first. Second one is capital T small p. Third one is small t capital P, and the fourth one is small t small p. Fine. Here again the same. Okay, small p, then small t capital P, and fourth is the small t small p. Fine. Now here you can see T is independent to, here the capital T is independently assorting with the small t, or the capital P, or the small p, or the t. Fine. Now this capital P is also independently assorting with the capital T or the small t. So now um, here, wait. So the capital T is independently assorting with the capital P and the small p, whereas the capital P is independently assorting with the capital T or small t. Therefore, we can say that allele of any gene, or you can say allele of one gene, can independently assort with the allele of the other gene. Okay, so that's why this law name is law of independent assortment. Okay, because independent assortment because here it is independently assorting. Here the allele of one gene is independently assorting with the allele of the other gene. So let me revise. So Mendel took pea plant for his experimental work and he gave some laws that was named as law of inheritance or Mendel's law of inheritance. He gave three laws. Okay. So he gave three laws. First name was law of dominance, second is law of segregation, third is law of independent assortment. Fine. Now, here in law of dominance, we are taking the pure lines. Pure lines means the homozygous form of the parents. Okay, homozygous means either they will be dominant or will be recessive. So parents, we are taking here the tall. Here, according to the law of dominance, Mendel stated that when we start with the pure line, it is the dominant which expresses itself. So here we are taking a cross between the tall and the dwarf, homozygous tall and the homozygous dwarf. Now, just then we are making the gametes, that is a capital T, small t and small t, these are the gametes. We are making the punnett square. Okay. So now here we are making the punnett square. Punnett square. This is the punnett square, okay? The graphical representation to give the possible genotypes of an offspring in a particular breed or cross. So this is the punnett square. We made this F1 generation. F1 generation is when we breed between the parents. F2 is when we interbreed between the offspring, okay? So we are making here the F1 generation. Now phenotypic ratio will be 4:0 as here all the offspring obtained is tall in nature. In the genotypic, that is the genetic makeup of an organism, ratio is 4:0 again. 4 is the heterozygous and 0 is the homozygous. Hence, here we see that the character or traits which is expressing itself is the dominant one. In the second law, the law of segregation, which is also known as the law of separation, here, during gamete formation, the allele of a gene gets separated. Now, whether we take the example of a homozygous or we take heterozygous, the alleles are getting separated during the gamete formation. And this allele is pure form of the allele for the next generation. Okay, so this law is also known as the law of purity of gametes. Now, the third law of independent assortment. Here it is best explained in the dihybrid cross or only explained in the dihybrid cross. Parents, here we are taking the two characters, capital T, that is the tall, capital P, that is the violet. We are just crossing it with dwarf and white. Okay, now these are the gametes obtained. If you want to learn to produce the gametes, just I'm explaining you here. Okay, this capital T with this capital P, then this capital T with this capital P, this capital T with this, and this with this. Okay, so we will get the four of the gametes. Okay, now we got the four gametes. We made punnett square, we crossed it and we got this punnett square as the F1 generation, and then we selfed it. Self means self fertilization. Now the parents here will be the offspring which is obtained in the F1 generation. So capital T, small t, you can say, see here, this we got. Now we are doing the selfing. Okay, now this again the four gametes we got, and this now we can obtain or we can observe here that this capital T is independently assorting with the capital P or the small p, whereas the capital P can independently assort with the capital T or the small t. Now we can say that allele of one gene can independently assort with the alleles of the other gene. Hence, this law was named as law of independent assortment. So, in the next video, we will be discussing some examples of the monohybrid cross, dihybrid cross, and the trihybrid cross. Thank you for watching.